The overall goal of this procedure is to isolate nasal olfactory stem cells from rats or humans in order to identify molecular anomalies or repair the pathological or traumatized brain. This is accomplished by first surgically collecting olfactory mucosa biopsies from the nasal cavity. Next, eliminate the olfactory epithelium and its epithelial stem cells to focus on ectomesenchymal stem cells in the underlying lamina propria. And then isolate those cells by dissociating rodent tissue or collecting outgrowing cells from human explants cultivated under glass cover slips. The final step of the procedure is to grow stem cell spheres, inducing differentiation into neuron-like cells. Ultimately, explants of the whole olfactory mucosa, individualized stem cells, floating spheres, or neuron-like cells can be used for identifying molecular markers in brain diseases or repairing the damaged nervous system.
We describe here a method for isolating human and rodent nasal stem cells from the olfactory mucosa. The main advantage of this technique over existing method is that the stem cells belong to the nervous tissue, are easily accessible in every living individual, and can be used for diagnosing or repairing the pathological brain.
As an ENT surgeon, I've performed hundreds of nasal biopsies for fundamental or clinical studies. The procedure is safe and can be carried out under local or general anesthesia.
I am a junior scientist with an expertise on the culture of human or rodent nasal olfactory stem cells. These cells display a high proliferation rate and an inclination to differentiate into neural cell types. They are proven to be efficient in animal models of amnesia and Parkinson's disease.
In advance of collecting the mucosa biopsies, have 3 35mm Petri dishes with DMEM Ham F12 culture medium readied in a clean culture hood. Now, after verifying via toe pinch that the rat has entered deep anesthesia with sodium pentobarbital, euthanize by decapitation, isolate the head, and remove the skin. Remove the lower jaw with scissors and, with the help of a rongeur, eliminate the facial muscles on both sides. Starting from the back of the incisors, using the rongeur, remove the bone covering the nasal cavity one side at a time. The olfactory turbinates come into sight as orange-brown organs located in the back of the nose. Delicately, discard the turbinates with forceps. Using a 26-gauge needle, isolate the olfactory mucosa lying on the septum by cutting the tissue along 3 lines: the arc of the perpendicular plate, the cribriform plate, and the ceiling of the nasal cavity. Collect biopsies on both sides and transfer them to a DMEM Ham F12 filled Petri dish. This procedure should take no longer than 10 minutes from the onset of euthanasia. Now, in order to remove the mucus, transfer the biopsies 2 times in medium-filled Petri dishes. In accordance with the relevant local ethical committees, an ear, nose, and throat surgeon should carry out this procedure, and every outpatient should sign an informed consent form. Using a 0° or 30° rigid endoscope, inspect both nasal cavities and assess the putative presence of polyps or any inflammatory lesion. Choose the best nasal cavity, taking into account the deviation of the septum. Using a cotton applicator, apply a local anesthetic, such as lidocaine with epinephrine, for 10 minutes. With a through-cut ethmoid forceps, collect a 2 square millimeter biopsy either at the root of the medial aspect of the middle turbinate or on the septum in the dorsomedial area. Here we show the biopsy from the root area. The olfactory biopsy is then transferred, using a sterile needle, into a sterile 2 milliliter tube filled with 1 milliliter of DMEM Ham F12. Tip the tube upside down to make sure that the biopsy is immersed in the culture medium. Insert the tube in a refrigerated container and transport it to the research laboratory. At this stage, the biopsy can be used per se for comparative molecular studies focused on specific brain diseases or processed for generating stem cells. Wash the human or rat biopsies in DMEM Ham F12. Incubate the biopsies in a petri dish filled with 1 milliliter of Dispace 2 solution for 1 hour at 37 degrees Celsius. Next, the human or rat olfactory epithelium is removed from the underlying lamina propria using a microspatula under a dissecting microscope with a diffracted inverted light. The olfactory epithelium is translucent over a black background compared to the lamina propria, which is striped orange-brown. Over a white background, the epithelium looks gray and the lamina propria brown. Once purified, transfer the lamina propria into a petri dish filled with DMEM-Ham F12. If the tissue is from a rodent, cut the lamina propria into small pieces with two 25-gauge needles. Then, transfer the pieces to a 15 milliliter tube filled with 1 milliliter of collagenase 1A. In the tube, using a sterile plastic pipette, dissociate the tissue. Then, incubate the tube for 10 minutes at 37 degrees Celsius. To terminate the dissociation, gently rock the tube and add 9 milliliters of calcium-free and magnesium-free PBS and centrifuge at 200 Gs for 5 minutes. Resuspend the cell pellet in culture medium and plate on plastic culture dishes. Now, if the tissue is human, slice the lamina propria into 3 to 4 pieces. Insert each strip into its own 2 cm diameter well in a 4-well plate and cover the tissue with sterile 1.3 cm diameter glass cover slips. Then, add 500 microliters of culture medium to each culture dish. For either tissue type, renew the culture medium every 2 to 3 days. After 5 to 7 days of culturing, the stem cells will begin to invade the culture dish, and after 2 weeks they should be confluent. When confluency is reached, passage and transfer the cells to culture flasks. To generate stem cell spheres, replate the cells at a density of 16,000 cells per square centimeter into pre-treated flasks. Every 2 days, feed the cells with 0.2 milliliters per square centimeter of supplemented medium. 2 to 5 days later, collect floating cell spheres and either replate or dissociate them before grafting in animal models of cell therapy. To differentiate the olfactory stem cells into neuron-like cells, cultivate the cells for 21 days in neurobasal medium containing B27, penicillin, streptomycin, glutamine, and glutamate. Then make medium changes every 3 days. Neuron-like cells should appear after 2 to 3 weeks. Five to 7 days post-plating, nasal human olfactory stem cells grew out of the explant and proliferated rapidly. Confluency could be reached within a week or 2. A key feature of stem cells, nestin expression, was evaluated. When grown on polyl-lysine with a serum-free culture medium supplemented with EGF and FGF2, the olfactory stem cells gave rise to spheres. When the spheres were plated in serum-containing culture medium, they gave rise to cell populations containing approximately 50% GFAP-expressing cells, up to 15% tubulin-expressing cells, and up to 5% O4-expressing cells. When the sphere-derived cells were grown in supplemented neurobasal culture medium, most of the population became neuron-like cells, expressing beta-3 tubulin. The same neuron-like cells also expressed MAP2. Thus, the fate of the stem cells could be modified.
Once mastered, this technique can easily generate hundreds of millions of stem cells in less than 4 weeks, allowing the clinicians to perform future autologous cell transplantation.
Nasal olfactory biopsies are very useful for identifying biomarkers and brain diseases. Recently, we have been successful in finding new candidate genes for autism.
We have already set up a bank of olfactory stem cells from autistic and controlled individuals, and it can be enlarged to other human brain disorders.